Thank you very much for having me here and for that introduction. It's a big room. You can have my seat. I'm not using it. First, the usual disclosures. I've started a bunch of companies. I consult for a bunch of companies. You probably shouldn't believe another word I say over the next hour. But actually, I'm most proud of, at the bottom right here, more than half of my graduate students now have started a company. And they do this with the most amazing platform in the world, which is open data. And basically the whole talk comes down to about 3 anecdotes of how we're doing science at Stanford using open data. What exactly do we do with it? And all 3 have ended up launching a company. So you're going to see exactly how we do it. All of our secrets are buried. So I'm very proud of this list. It's something to be ashamed of. I think we kind of get it at Stanford that if you want to change the world, you can't just keep writing papers about it. You've got to do something about it. So it's no surprise to you all that we're in the middle of this data deluge. I still show this cover from an economist, I guess it was 3 years ago, that said at the time that we as a human species generate 2 zettabytes of data each year. You can see the metric prefixes there if you forget what a zettabyte is, kilo, mega, giga, tera, peta, exa, zettabytes. And of course, now we're generating 4 zettabytes or 8 zettabytes. Because of course this is growing exponentially. This is no mystery to you all. You also all know that probably all of these zettabytes involve videos of cats playing and kittens on pianos, right? Perhaps not the most scientifically relevant, but still entertainment value nonetheless. But of course there is scientific data out on the internet as well. We know the Large Hadron Collider generates petabytes of data, right? The NASA has these digital sky surveys that generate petabytes of data. So many pictures of the sky that there aren't enough astronomers out there to look at it, so they just crowdsource that to the internet. You can go and pick some pictures and decide, is this a planet or is this a galaxy or a star? So we're used to this, and of course we have this in life science data as well. And what we're talking about is big data in biomedicine. I don't know if you know or hate this word, big data. I don't care particularly, buzzwords come and go. I think we all know what we're talking about here. Harvard Business Review had this great article if you're looking to convince the business folks, the people who make money in your institution, about the next big scientific revolution. Science, that's a big thing. They're talking about paradigm shifts, moving from theory to experimentation in the 1600s to computers and simulation in the 1900s, even simulated mice like model organisms, and now data mining. Nature says it's shameful if we don't use the data out there. Science says we need to make the data maximally available, and even the Lancet says if we don't share research data, it's an impediment to public health. Now, what are we talking about here? What am I going to be talking about? It's data from devices like this. And so on the left here is what we call a mathematics gene chip. Some of you might be familiar with this type of device. But this is an example of a high throughput measurement modality in biology and medicine today. And because I like to bring prompts with me, and because data can sometimes be seen as too ethereal, this is actually what one of these looks like. Okay, so you can actually see. It's kind of neat. It fits in your shirt pocket, right, if you have a shirt pocket. And this is amazing. It's an amazing device, right? With this one chip, it's the size of your thumbnail. Here's the size of a thumbnail here. And this lets us quantitate every gene in the genome. Quantitate. You can use it for spelling differences. We'll talk about the spelling differences. This lets us measure every gene in the genome, how much is present on that tissue or sample you put on there. And then when you get tired of measuring them one by one, they even come in this famous 96-well format. We love 96 wells in biology and medicine. Because you can find pipetters that go this way or that way, and each well here now is one of these chips. And this is an amazing device. It looks like a computer chip. This company, Affymetrix, is just one of many companies that make these kinds of high-throughput measurements. They happen to be based in Santa Clara. It's kind of across the street from Intel, so I guess they make it look like an Intel chip. And this is amazing, but we have to remember, this is already a 15-year-old invention. As amazing as this still looks, as amazing I think about it, I still grin when I think about this. We've had these now for 15 years. Everyone uses these. And what's happened is because everyone uses this, everyone started writing papers about this, right? We all know this. Here's a list of genes I've changed in that cancer. Here's a list of genes I've changed in heart failure. Here's a list of genes I've changed in that disease, and this disease. And what happened in the early 2000s, 2001, 2002, is everyone started submitting publications. And then the top tier journals start to realize, we don't know what people are doing with this data. And started with articles like this, show me the data, right? Charles Perrault saying, there's a lot of data sets being analyzed, where is the data? This was back in 2001. And that led to the top tier journals starting to require that if you want to submit a paper to those journals, you need to get an accession number for this type of data. And the precedent had been set almost 30 years earlier, going back to GenBank, right? DNA sequences. Nobody wants to type in DNA sequences again from a figure in a paper. You guys submit that electronically. Now you got to submit this kind of data electronically. You got to submit a lot of data electronically. So almost every one of our academic institutions uses these chips. Everyone knows what to use them for. Very few people realize how much data is actually publicly available today. As of August 2012, Nature did an article about me and a bunch of others using this data. As of August 2012, we hit 1 million of these chips. 1 million of these chips publicly available. Okay, that's up from 0 in 2003, 1 million. And I'll show you where it lives in a moment. It looks kind of sad, though. It looks like it's kind of slowing down, you know? And then you realize they only counted half a year, because this came out in August, right? And you realize, oh my goodness, it's still doubling every 3 to 4 years. It's finally slowing down compared to Moore's law. Slowing down from Moore's law, okay? We were already way faster than Moore's law. And actually, if you look today, the data lives in 2 big houses. One is the Gene Expression Omnibus run by NCBI and NIH. The other is the European one from EBI, Array Express. And they both have some overlap, but actually to get rid of the overlap, we're already over 1.25 million. Okay, 1 million was like 2 years ago. We're already at 1.25 million microarrays. And if you don't like these chips, it doesn't matter. Every repository is like this in life science. Not only does every repository grow at an exponential rate, the number of repositories is growing at an exponential rate. So that's a double exponent. If you think, well, which is still an exponent. Think about that, right? So what does this mean for you or me? Well, you know, some of you see my videos on YouTube, so I'll just bring this all the way down to home here. Here's a search box, right here. You can search for any disease. Now, why you? A high school kid today who needs to do a science fair project in 11th grade or 12th grade, she can type in something like breast cancer, and now find and download about 40,000 samples of breast cancer for her science fair project, about as easy as she can find a song on iTunes today. Right? 40,000 samples. All samples, most of them are human, some mouse models, some rat, some dog, some viruses, publicly available. She's starting an experiment with 40,000 samples. If you haven't figured it out, what's so nice about 40,000 samples? That's more samples that that kid can get to than any breast cancer researcher will ever have in their lab. Right? So whoever gets to the middle now has more samples than anyone contributing the data. Right? That's going to be the rule of thumb. You always have more data than anyone else in the field if you know how to get to the repository. Now 40,000 samples is kind of interesting. Actually, the number I like is this one. 1,500 labs have contributed data to this repository. 1,500 different labs or teams. Right? What does that mean? Let me explain it to you in a different way. Let's say I was coming to you writing a brand new grant to study breast cancer. And you're the study section, you're the review panel. And I propose to you, I'm going to get 1,500 of the best labs in the world who study breast cancer to run the best technologies in the world, to get their best characterized patients, to collect all that data, and in my grant, they're actually going to give me all that data for free. Okay? If I propose this in a forward direction, right, getting my friends on the internet help, they call it crowdsourcing, right? If I proposed the crowdsourcing in a forward direction, you would laugh me out of the room. How would you get 1,500 people to give you new data for free? Yet, there are 1,500 teams contributing the best measurements they have available for free. So the other way I like to think about public big data is actually retroactive crowdsourcing. Okay? The crowd is there to help you, and the crowd doesn't even know it. A crowd is there to help you, and you don't even know it. But that's data. That's public big data today. And if somehow you're not happy with 40,000 samples, wait a couple years, it'll be 80,000 samples. Just in the course of this hour, we'll add a couple hundred samples to the repository, doing nothing here, right? So at a certain point, you start to realize, maybe I should start to use this data, right? Not just kind of count the numbers, you know, I want to start to use this. By the way, if any of you have any doubts a high school kid can do this, here are the high school children who do it. All right, so some of you know Brittany Wenger. I've never met her. She's in Florida. At 17 years of age, she actually got public data, immunohistopath data, pictures, and came up with a neural network to diagnose breast cancer. She won top prize in the Google Science Fair competition. And then the following year, last year, she was in the newspaper for doing the same thing again now for leukemia. So she comes up with diagnostics this way. Some of you know about Jack Andraka, a kid comes up with pancreatic cancer, paper-based diagnostics, paper ELISAs. So high school kids can do it. And of course, what I mean is a high school child can do it. Probably you can too, okay? And if you're not interested in anything I'm saying here, get your kids to do this, okay? Because we need more people like this, right? Coming up and making great. If RNA is not your thing, DNA is your thing, if you're into cancer, here's another big repository, Cancer Genome Atlas. You know, NIH, a lot of these funding organizations fund these big science projects. A couple of scientists involved get a paper here, a paper here, a paper here, and here's all the rest of the data. And then they move on to getting another grant to collect more data. And nobody's around to really analyze the data that they've already collected because people think, oh, it's stale, it must be, oh, it's free and on the internet, it must be valueless, right? That's ridiculous. Amazing data. I think the data we create and export to each other is the most amazing creation that we can export, right? It's better than oil, it's better than water, right? My using the data doesn't take it away from you. You can use it too, right? And we can create value on this. It's the most amazing concept, this data. Any kind of cancer here, 20 cancers, 20,000 samples publicly available. If you're into DNA, the DNA repositories are growing even faster. And here, you know, DNA we're a little bit more finicky about, right? Because you can identify a person. We have HIPAA, we have rules like that about personal health identifiers. So here you can't just click to download the data. You gotta actually get permission forms from your institutional ethics board, maybe, for some of these. But here the rules are even more severe. If you're funded by NIH, you're collecting DNA data, you gotta put the data in this repository before you publish it. So to give them a chance to write a paper, we got embargo dates. Of course, by the time you get to the embargo date, most of them have expired. So why would I deal with this extra headache for DNA? I just want to point out here in the middle, at the top here, the entire Framingham Heart Study. Okay, what is so interesting? The Framingham Heart Study is a 30, almost 40-year-old study, longitudinally looking at the entire city of Framingham, which is just west of Boston. It's the reason why we know the word cholesterol. Okay? It's kind of neat now. I can get the DNA of, what, 14,000 people. Actually, more interesting is you can get 30 years of measurements of these people, right? Who were their friends? Where did they live? And of course, all the medical and height and weight measurements waiting for you. If you think they've made every last discovery in the Framingham consortium, you know they haven't. There's so much more. You could go and say, hi, let me go look for every patient that got cancer of this kind, and let me just see 10 years earlier. Could I have predicted who was going to get cancer? Now, all that data is just sitting there. And that's one of thousands of studies here. That's the nature of data today, so much of it. On the drug side, it's even more amazing. If you're into chemicals and drugs, I don't mean if you take drugs, if you discover drugs, right? PubChem, if NIH funds you to do drug discovery, drug screening as an academic, they'll give you the dollars, but you've got to deposit your data to PubChem. So the way to think about PubChem is a big grid of 120 million chemicals and compounds, so it's the columns, 740,000 screening assays or bioassays. If you do that math, that grid is about 100 trillion little boxes, of which 1 billion of them have some measurement made by some scientists in the last 10 years. Now that's just amazing to me. I'm willing to bet, I've said this in talks before, I'm willing to bet any of you a beer, this beats what any pharmaceutical company has. Just publicly available. Actually, I can easily beat you, I bet you a bottle of wine. This beats what all pharmaceutical companies have combined. Because this is doubling every 2 to 3 years again. And as you can see in a couple slides, nothing in pharma land is doubling anymore, okay? They're laying off these kind of discovery folks. And you can get to all this data today without a user ID attachment. Okay? Completely open. Every future drug is probably one of these columns here. And it's already been screened for something, waiting for some scientist to come and get to this data. And it's only going to get better. Some of you know the White House made this announcement last year around this open access thing, where publications have to be made publicly available. If I write a paper, if I'm funded by NIH, I have to make my papers available within a year. Those of us funded by NIH, we kind of already had this rule, it wasn't a big deal, but now it's Department of Defense, Department of Energy. Anyone funded by the government as a grantee has to make their papers available. It's kind of interesting. At the bottom here is the fine print. They said, oh, by the way, you gotta make your data available too. Not just the papers. That's just going to be a tsunami of data. Again, more and more coming because of mandates. It starts with the publishers, actually starts with the editors, goes to the publishers, then goes to the funding agencies. It's a constant trend you're going to see again and again. So what do you do with the data? So let's just give you a couple of anecdotes here, see what gets you interested. First, we start to realize my first big NIH grant was go get every human disease studied with these microarrays. Right? We have all of these microarrays, got a million of them. Go get every human disease that's already available. And they're structured like this, experiments where we look at disease individuals and healthy individuals, disease healthy, disease healthy, and we measure genes. I'm not going to go into any kind of molecular detail, you can just see they're color-coded, and we calculate the differences. The signature is a difference between healthy and disease, But why stop at one? Actually, you end up with hundreds of these. And so this one paper actually drives a lot of the themes that we talked about in my lab, and I'm going to just spend a moment on this slide, because this might be a great take-home point for you all. This really drives the things. This paper came out in Nature Methods, April 2009, Questioning Standardization in Science. We're in this huge trend right now that science should be reproducible, right? If you get an answer, you should have the same answer, right? And NIH is worried about this, and National Academy of Science is worried about this. Let me just point out this interesting paper. This paper was written by the mouse psychiatrists. You think about those two words together, right? The mouse psychiatrists. And so they study anxiety and depression in mouse models. Of course, they're trying to get to genes that have relevance to humans, right? And for 10, 20 years, decades, a West Coast lab would get a gene, an East Coast lab would get a gene, and they would get completely different genes. I said, we need standards in science. We're all doing different experiments here, right? None of us getting the same answer. So I said, okay, this is what it means to study anxiety. This is what it means to study depression. You got to put a mouse in this kind of cage, this kind of maze. You got to do this exactly the same way. And what happens in their field? They all start to get the same answer. You say, great, science is reproducible. West Coast lab is getting the same answer as the East Coast lab. Now these guys came along and they said one interesting thing. They said, well, what happens if you change the strain of the mice, put 2 mice in the cage, give them something to play with? None of the answers hold up anymore. They've done what we call overfitting the model. They got such a detailed model, of course everyone gets the same answer, but they're least relevant to the general case. This group says, you know, this is going to be controversial, but why on earth do we standardize things in science? We should have 100 or 1,000 good ways to do this experiment. And then see what answer sticks at the end, right? Maybe more than half the people get the same gene or the same answer, right? It is actually irrelevant if we all get the same answer by doing the experiment the same way. We want 100 different ways to do this experiment. And what better way to think about doing science this way than playing with the data people have already generated and deposited into the repositories. I don't exactly care how you did this experiment. I'd rather have 100 mediocre ways to do this experiment than one person who says they're doing it the right way. I don't believe you. I'll take 100 experts that think they're doing it some decent way and then figure out what's in common. It's a long introduction to talk about how we do this in the lab. So really, we have hundreds of experiments for hundreds of diseases now. And what we do is we start to turn our crank, we start to get some ideas. Our first big idea we realize is there's some genes that are in common. Everyone seems to say this gene is up, this gene is up. What we realize is those genes are great, but that's a great academic paper, but we can start to actually think about measuring those genes in patients for this disease. And that becomes a blood test, or what they call a biomarker, a diagnostic. If everyone keeps saying this gene is up, let's make this into a diagnostic. And so we turn our crank, we have a disease called acute myeloid leukemia. It's not a great leukemia to have. And we come up with such a diagnostic. We say, oh look, it looks like we're predicting this protein seems to be up in the blood. So we go to our cancer center at Stanford. I love them, I know them. Do we have any saved blood on patients with acute myeloid leukemia? They said, actually, no. We saved the cancer cells. We never saved the serum or the plasma. It's too many freezers you need for that. We said, okay, now what do? We're stuck. So what does a computational person do today? We have public data. We got these predictions for blood tests. We just Google for it. This is the new world. Conversant bio. I see pictures of blood tubes. Browse by disease. What is it that you wanted to validate tonight? Bladder cancer, brain cancer, breast cancer. Okay, here's leukemia. What do you want to buy today? CD19 positive B cells from bone marrow leukemia, CD45 positive cells, mononuclear cells. Page 2. Okay, peripheral blood from leukemia. $55 each. How many samples do you want per patient? Add to shopping cart. This isn't made up, this is real. What is this? The hospitals in the middle of the country that don't have medical school nearby. When you order a blood test as a physician, the lab holds it for 48 hours, waiting for you to add more tests. What happens at 48 hours? They need the freezer space. It's on its way to the trash. If something magically legal happens in the United States when samples are on their way to the trash, if you strip off the identifiers, it becomes now a discarded sample, and companies come in and curate these samples and then resell them for research use. Okay, and I have this now in 48 hours on dry ice. This is not the hard part of it. Now I'm saying it's magically legal. I didn't say this is ethical, okay? Ethics is a region by region thing, okay? It's a community thing, right? They get away with a lot in Boston, we can't get away with it in Palo Alto, I'll just say it that way, okay? So that's a region by region, but it's magically legal to do this, okay? Ethics are a little bit beyond that. $55. Some of you might realize that's cheaper than my own institution would take to actually save the sample. I'm just showing you the top Google hit here. There's hundreds of companies that do this. For pathology samples, if I'm so moved that Steve Jobs died of pancreatic cancer, I want to study pancreatic cancer. Here, I can buy 100 patients' cancers. 42 of this kind of cancer, ductal, 3 adenosquamous, 1 islet cell, 6 metastatic. All these pathology samples were read by a pathologist. They got an extra sample and made a core, a full circle, put them on a glass slide here. $395, and again, a little picture of a shopping cart. 100 samples of pathology. 48 hours, comes right in the mail. Okay? And that's how we can test and validate blood tests, but diagnostic biomarkers. So we got papers published on cancer biomarkers. You can see all the cancer samples we can buy. Came up with a blood test for transplant rejection, so when you get a new organ put in, if you're rejecting, you wake up one morning, your body you don't feel any pain, but your body says, this kidney wasn't part of me, I gotta get rid of this thing. You wanna look out for that because you need to go on medications. We have blood tests for that. But my favorite one is actually this one. The disease called preeclampsia, which nobody really remembers preeclampsia, except it was on Downton Abbey, so now everyone knows preeclampsia. Okay, it's back. It's always been there. This is how husbands lose wives, even today. Okay, it's by preeclampsia. It's a high blood pressure problem during pregnancy, and when it can lead to eclampsia, which is seizures and death. It still kills a lot of women around the world. It leads to newborns being born very early, and it turns out there's no blood test for this. And so we said, let's come up with a blood test for this. So what do? This is Linda Liu, grad student in my lab, Matt Cooper, Bruce Ling, my great collaborators here. You just search how many data sets on preeclampsia, preterm, preeclampsia. All of these have some experiment in some tissue, human or mouse, on this. You gather all the data, figure out what's in common. These are all up, these are all down. And here you go, funded by March of Dimes. Here's a biomarker blood test. This is lower in women who are going to get preeclampsia. By the way, we start by just buying these samples off the internet. It works. Let's go to our own patients, saved samples, and let's start cohorts moving forward. Getting the samples is not the hard part anymore. And actually getting the idea isn't the hard part if you're a data scientist, if you know how to do this. And of course, in the Stanford way, this spins out to be a company called Carbon. Okay? This is a brand new company. Diagnostic, 18 months from computation, a company funded with $2.2 million by angel investors. Okay? Science doesn't end with the company, the science launches with the company. It's another thing to realize. Okay, the science continues, but it's going to get into the patients here. And you can see, $2 million. Need a diagnostic, seed angel investors come in, 18 months. Start with public data, launch a company. That's the first thing. Let's get into drugs, therapeutics. Bigger problem. How much does it cost to make a new drug? Usual answer people say is $1 billion. Everyone says it costs $1 billion, it takes 10 years to come up with a drug. That is a huge underestimate. Okay? Billion is nothing. Okay? How do you calculate this number? Very simple formula. Pharma R&D dollars divide by the number of drugs. Okay? Very simple equation. R&D dollars divided by the number of new drugs. If you do that equation, you start to realize it costs between $4 billion and $12 billion to come up with a new drug. Not sustainable. And the reason this keeps getting higher is because the numerator is growing, denominator is not changing. Okay? 4 billion to 12 billion. It would probably be tolerable if this was actually working at that level of expense, but it's not working. So much so that we're running out of drugs to sell. And so what that's going to lead to in 2018 is called a patent cliff. The pharmaceutical company has so many patents coming off patent to be generic, which is a good thing, right? Patients need to be able to afford these medications. But they don't have enough new ones coming out that the entire pharma industry is predicted to lose $250 billion of revenue that year. Not $250 million, $250 billion. The entire pharma industry will lose that year in revenue. Now, you might love the pharmaceutical industry. You might hate the pharmaceutical industry. I'm not here to pass judgment. But if the entire industry is losing $250 billion, I kind of wonder where the research and development is going to happen in the future. And I kind of wonder if it's going to fall to all of us to figure out these drugs. And if you look at the pharma model today, they're really into in-licensing. They're really into getting rid of their own R&D folks today. So it's going to be up to all of us, including us in academia, to come up with some of these new drugs, to validate them as far as we can. What better way to do this than with public data? And that's what I'm going to show you. So now, instead of just having disease individuals and control individuals, we also have samples, cells, treated with drug, without drug, with drug, without drug. It should be a PubChem repository with trillions of little measurements, billions of little measurements. So I've got with drug, without drug, tested on cells. I have disease samples with disease, without disease. And why don't I just put those two together? And the reporters for this call this Match.com for drugs. Now, some of you from out of the United States, so if you don't know, Match.com is what we use for setting up dates and maybe you get married this way. And what's the famous saying? Opposites attract, right? So that's what we're going to do here. If I've got a disease that makes this gene go up and this gene go down, and then I can find a drug that can make this gene go down and this one come up, maybe there's a match in heaven there, right? Made in heaven. So I'm going to match up these to see if drugs that reverse the effect of the disease. And you can imagine 2 genes, imagine 20,000 genes. You can use correlation coefficients, a lot of ways to do this. I'm not going to get into any formulas here. But then we get stuck again, right? Then we get stuck. I got this idea. Here's a drug, seems to work on this disease. How am I going to test this drug? I can go find my friends. I can go find my patients. They say before you put in a patient, you got to test this in an animal model, like a mouse or a rat. How am I going to get that done? I'm a computational guy. And now, when I really, really need to get a drug tested in an animal model, I can go to assaydepot.com. A few people chuckling realize this kind of sounds like Home Depot, okay? It's huge, again, for the outside people. It's a huge store across the United States. There's like 2,000 of these stores. Everything you want for your house is there. So here are my aisles in my store. Biology, chemistry, DMPK stands for drug metabolism pharmacokinetics. If you don't know what that stands for, don't go in there. Pharmacology and toxicology. Let's go to pharmacology. What type of mouse do you want to run today? Bone, respiratory, dermatology, diabetes, urinary infectious disease, inflammation, neurology, cancer, eye, ear, pain, respiratory. I'm a diabetes guy. Let's say I just click on that. Okay, I want to run a 16 mouse experiment. OB diabetes mouse model. Okay? You read here, it says this mouse has been eating a lot and getting diabetes since 1951. This is a standard mouse that gets diabetes, and a lot of people test their drugs with this. 16 mice. Do you want 2 groups of 8? Do you want 4 groups of 4? In 28 days, they'll test all the insulin levels, the blood sugar levels, and they'll give you the results. You don't have to tell them what drug you're testing. You can send it all blinded. You can order the drugs from one company, get this from this other company. Now, what am I covering up? The price. $9,000 for this mouse service, 9-week turnaround time, and again, add to shopping. Entire mouse model you can buy off the internet with a credit card today. Okay, off the internet. Now here's the real world. Okay, someone with really sharp eyes see this is run in China. Okay, some of you may have a problem with that. I don't have a problem with that. But what happens in reality is when you hit add to shopping cart, you're presented with the 133 companies or labs that actually are willing to offer this service to you, including Maryland and Wisconsin. Wherever you want in the world, there's going to be a company that does this. Outsourcing doesn't always mean offshoring, okay? But companies like AstraZeneca have curated all of these companies. They give it to you in a clearinghouse. Now, maybe you want them to be USDA certified. There's only 4 of them. Click here. ISO 9001. Click here. You only see 7 of them. AAA line, 28. Whatever certifications you want, you click, and you're only going to be presented with those that are so certified. Oh, how I wish it were that easy to pick the right collaborators at Stanford. And I know it's not some random grad student doing this mouse experiment, but someone with some certification, right? And now you're wondering, boy, you know, all that data and computation, we love that stuff, but this seems like the most important part. This is the part that tells if the drug is working before you put it into humans. How do you trust any one of these companies? I'll simply say that if you really, really need to know whether this drug is working in this mouse, simply order 2 of them from 2 companies. I don't know, $8,000, order 10 of them from 10 companies, right? I can easily convert the quality issue into dollars now, right? I want really more quality, order more independent teams to do this. Let me just put this in perspective for a second. How many mouse biologists, when they get a mouse experiment working, do they go across the hall to get a completely independent team of people? You know they don't do that, okay? So I will always win the quality argument now, because I can just hire more people to do this experiment independently, right? Quality is a non-issue anymore here. I just get more people to do it. And that's what we do. Our first big example was a seizure. So, by the way, I'm making a leap of faith here. I made a leap here. You know, we can do this for new drugs, where we have a lot of fun is to do this with existing drugs. Well, a story they call drug repositioning. Well, why do we do drug repositioning? Because these are drugs already in the clinic, already cheaper, and we can get them to patients faster. And so our best example of this, our first example, was a seizure drug called topiramate, epilepsy in the brain. We predicted using public big data would work on Crohn's disease, a disease where you're kind of attacking your own intestines. And what we learned is you give TNBS, you know, up where the sun don't shine on a rat, and it gets Crohn's disease, and here's the steroids, it works well, and here's the epilepsy drug topiramate, it treats it just as well. We get this great data, we submit it to Science Translational Medicine, and the editors love it. We're working with Mohan, Shinoy, Jay, Pashrika, but they said, you know what, can you get better quality data on this? And what happened is, I love Mohan and Jay, but they were giving us iPhone pictures of rats, okay? Now we have fancy microscopes at Stanford and all this, but they were taking iPhone pictures, not the highest quality pictures. So we said, Mohan, Jay, we love you guys, but we gotta outsource this, okay? So we go to Assay Depot, we find 100 companies willing to do the TNBS experiment. We pick one up in Berkeley, because they're having a sale. Literally, their discounted price includes serum cytokines like TNF-alpha levels, things that would be useful to measure in the rat blood. And then we picked one company out in Cambridge, Massachusetts. Freaking expensive, but the only ones in the world who knew how to do a rat colonoscopy. These are my famous rat colonoscopy videos. This is a normal rat. This is a rat with the Crohn's disease. It's a TNF. You see a little bit of redness there. This is a rat with Crohn's disease, with the MA, and with the seizure drug. I got 40 megs of video to show you on the paper. Now, for the youngsters who have not had a colonoscopy, let me just tell you why the rest of the people are groaning here, okay? Let me show you the geometry involved. This is a colonoscope, and this is a rat, okay? This has got to work. This is exactly what you want to outsource to someone to figure this out, right? So I'm not just outsourcing to save money, I'm outsourcing actually to get the best damn data in the world. No one at Stanford knew how to do a rat colonoscopy, right? So it's not just about saving money, it's about getting the right data here. That's what we're talking about. Actually, this is my favorite story. This one just came out a couple months ago. Again, great collaboration, Julius Anonymous, Joel Dilly, my lab did the work, and maybe John, Joel Neal's involved now. This is a mouse that gets lung cancer. Now, the rest of us humans have to smoke to get lung cancer. This mouse, you knock out these 3 genes, it gets its own lung cancer after a couple weeks. And on the right here is the same mouse treated with an antidepressant, okay, called imipramine. It's a tricyclic antidepressant. We don't use this drug anymore, okay? We have serotonin reuptake inhibitors like Prozac and others. This one has some kind of minor side effects, kind of makes you sleepy, and in some people widens your EKG, it's called QT prolongation. It can lead to an arrhythmia. Actually, neither of those sound as bad as having lung cancer. And this lung cancer is melted away. It's gone. It's melted away. Okay, this mouse doesn't have it. Why am I showing you this one? We got 10 stories like this. Why am I showing you this one? Because we start with public big data. We get this to work. We make the prediction. We test the cell lines in a dish. I'm not going to show you that. We get the mouse, you know. But of course, a lot of people cured cancer in mice for years, right? But that was enough for us to get ethics board approval, IRB approval, and 15 months after the computational prediction, we've launched a clinical trial already at Stanford on this drug. Now, I don't know if this drug is going to work in actual humans. I don't know. It's kind of like we are already winning because we went from public data to a trial in 15 months. Cost of the trial, $25,000, not $25 million, okay? It's a seed grant. It's never, oh, you got the drug already in the pharmacy, you don't even have to buy the drug. It's pennies per pill. So I don't know if this is going to work, but we got to launch more of these, right? This is what we got to do. It's what patients really want us to do. So that's where I think big data can get to in some ways. So I'm going to skip ahead a little bit in the story here because we're running out of time. But oh yeah, I got to show you this one here too because it involves me. Stanford, they got to do. They got to launch a company, right? So we get the platform, public big data. We can find new uses for drugs. Launch a company called Numeti. They already get a pharmaceutical deal. What does a pharmaceutical deal mean here? You know about phase 1, 2, 3 trials. Let's pretend a mouse is phase 0. The drug companies actually wanted a phase -1, just a computational prediction. Oh, you're predicting drug works? We'll license it from you. We'll take it all the way to patients, but we want that time. Phase -1, even before it even hits the mouse. That's really where the field is moving. They're looking for answers. $250 billion. You got an idea to come up with drugs. They're willing to listen to you right now. And that's how you launch a company. $3.5 million in venture capital funding, and the science continues. It doesn't end when you start a company. It continues in a new way with the company. Because I want to leave time for questions, I'll just skip ahead a little bit here. Let me make sure I know what I'm doing here. All right, let's just get to this. What do I think is the next big public data that's going to come out? This initiative that I got involved with called Import. And so Import is a 5-year repository. It's been running for 5 years. I took it over just last year. I think the next big data that's going to come out are clinical trials. What's a clinical trial? This is why it's costing $1 billion to come up with a drug. These are the most expensive experiments in the world. A diagnostic or a device or a drug. Company pays $100 million, $200 million to see if the drug works. And you know what happens? 50% of the trials fail. And when it fails, they don't even write a paper about it. Forget about letting the data out. The culture is set up that they don't even want to write a paper about the failure. So what does the rest of the world do? They kind of repeat the same mistakes again. We're not learning the science, right? So I think the next big push, whether it's going to be it's going to start with the journals, then it's going to get to the funding agencies, it's going to push for raw clinical trials data coming, not just the summary stuff on clinicaltrials.gov, but even the raw data that in the future we can do meta-analyses, reanalyses of data. Where is the drug working? Can I figure out which patient subset? So look, my eyes are on clinical trials data. I'm betting my lab on this because I think that's going to be the next big source. So we've already released 100 raw clinical trials. If you're curious, go to this website. All right, let's close out with some thoughts here. In Silicon Valley, we're so used to the kids today starting amazing high-value companies in their garages and dorm rooms. The famous examples, of course, of Apple and HP started in dorm rooms, garages. We had Yahoo, Google, Facebook starting in dorm rooms. Actually, I think the future is going to be about kids starting the next Genentech, the next Amgen, you know, in their garage. When you go home today or tomorrow at the end of this week and go stand in your garage, you already have 1 million microarrays and every mouse model in the world available to you with a credit card. Okay, maybe you need a decent credit limit. That's angel investors are for that, seed investors. Maybe you work with each other to get your credit limit higher. But I'm actually tired of kids coming up with yet another way to share pictures with each other. I think kids in the future should be coming up with new drugs for cancer and diabetes and all the rest. And of course, I got to show my own daughter at age 6 playing with DNA. Our museum's here in San Francisco. This is at the Tech Museum in San Jose. If you ever have time, go to the Tech Museum. If you want to play with DNA on the 3rd floor, they show you, even as young as age 6, how to move jellyfish DNA into a bacteria. They show you the whole experiment, and you don't even have to go back to see if it works. They'll take a digital picture. You can look in the next morning and see if your experiment worked or not. If you don't have a museum like this near you, you should be involved with making sure one of these comes about near you. Because we don't need more ways to share pictures. I love the tech industry, of course. But we need more of these kids thinking about this in life science, and digital data is an exciting way for them to get it. It sure beats dissecting frogs with formaldehyde, right? Start with 30,000 samples of breast cancer, come up with something cool, right? That's what we should be communicating to the kids. So take-home points, the patient samples, molecular, clinical, even epidemiological data already exist. They're already publicly available. They're already there to make an impact across medicine, but it's up to you to make this impact. Waiting for the perfect tools, perfect infrastructure, perfect data, perfect adaptations is waiting too long. It will never be perfect, but it's pretty damn good enough to do amazing things with it. And it's always getting better. If you wait for perfection, you're just going to miss the whole thing. Need for perfection sometimes hides data. The scientists love not to share the data. Oh, you mean you're still working on your ontology for this? Great, I can wait another year before you make me share this data. Right? I'll rather take free text from them, okay? No excuses, share the data here, right? The hardest part is we need investigators, and whether it's citizen scientists or tenured scientists, to come up with this career-changing move to use public data. It means trusting other people's data. It's a hard thing to do for some scientists. I don't trust any one of these data sets I use, but I trust what's in the collective wisdom across hundreds of them, right? Wisdom of the crowd here. And it's a new way to do science. For some scientists, it's hard, but we need to find those folks and keep them in biomedicine. Couldn't do any of this without a long list of collaborators. Specifically here, Alejandro Superintendent on the cancer side, and Jay Prashvik and his team on the GI side, and a lot of others on the diagnostic side. Couldn't do any of this without a long list of support. Lucille Packard Foundation, Joel Self giving me generous endowment support. A lot of this funded by 17 NIH grants from these 9 institutes of NIH. These 4 give me more, these 5 give me less. I still love them. HP, a lot of other companies, HMI. I always thank my admin tech staff wherever I go. I never get a grant or paper out the door without them. I always thank my daughter for giving me inspiration, and my wife, a molecular biologist, biochemist, who reads every major paper and grant that comes out of my lab, who starts companies with me now, and lets me go all over the place to give talks like this. Thank you very much. I left 2 or 3 minutes for questions.
Absolutely. I'll leave it so.
It's a question in the middle.
Okay.
Right.
I'll repeat your questions. You know.
There is a lot of effort in the policy arena and in institutions and in setting up incentive structures to encourage scientists to share data.
That's right.
How are we going to get to the point where we have incentives that motivate scientists to borrow data, reuse, and reanalyze data?
That's a great question. So we are getting to incentives, but I'm still arguing that we've got a long way to go. We have some incentives to share the data, whether it's publications, citation metrics, alternative types of impact factors, citable DOIs for data. All of these things seem to be working. Plus, there are a lot of sticks involved too. You must share your data, otherwise you don't get renewed, you don't get a grant. What about the reverse side of using the data? We're starting to see some tangible ways that this would work. For example, NIH now has, a couple of institutes have these grants called secondary use grants. So for example, the National Eye Institute, Allergy and Immunology Cancer Institute, if you can reuse this Cancer Institute funded data set for something else, they'll give you the support for that. Now it's tiny dollars compared to the generation of the data, but in some ways it's still starting. What I'm going to argue though is the incentives are there. I'm trying to show incentives on the reverse side, the pull side. Look, you can start a company in this too if you want, right? Even investors are willing to put dollars into this if you can come up with these diagnostics. Now, I think for some disorders, some ways, this is the best way to come up with drugs and diagnostics in some ways. It's not just one way, but I'm arguing this is the best way. So I think the incentives are there. The funding agencies are just starting, okay? I think people are starting to showcase reuse data. These are, heroes in some ways. But I'm trying to also show the real use cases. It's not just a paper for you, it means even a company, it means even outside dollars coming in to keep the science going. So I don't know if that answers your question, but I think the incentives are coming a long way to go, long way to go. Other questions, thoughts, issues, challenges? There's a question all over there. Go.
You implied that some
Wait for the microphone to get you.
They want to record you, so.
You imply some issues of ethical concerns at one point, and certainly just ordering up mice to be operated on in the UK, there are certain restrictions on how many mice and whatever, and ethical approval is required at all stages. Do any of those sites require an ethical approval document to be sent in?
Yeah, so great question. So do any of those sites require an ethical approval document to be sent to them? The human sample side, absolutely not. So these are discarded samples. It's up, you have to agree not to try to identify these people. They're much more worried about you trying to figure out who these patients are, okay? What you do with those samples, they're on the way to the trash, so that's right. For the mouse side, it's up to you to get the ethics that you need for your institution. Not everyone needs those rules. So the easy way out though is, for example, if your institution requires the work to be done in a triple ALAC, it's AALAC, it's a accreditation organization that actually shows that the mice who perform, the experiments are done perform right. It costs more for these organizations to get triple ALAC certification, but you can just click there and just that way ensure that your work is only going there. A lot of times, let me put it this way, right? So let's say you, as an academic, are writing a grant, but the mouse work is going to be done in the United States. Let's say it's a subcontract, commonly done. Typically, it's up to the receiving end of your dollars to get the approval there. You don't do duplicate work. You might have a prediction in the UK to send the work. It's usually the other side. You want to make sure it's dotting the I's, crossing the T's. Same thing for these companies. You can go to their websites, you can see if they're approved or not, or just click to order them. So you're absolutely right. So it's going to be a different scale. All I'm saying, though, is paperwork is not the barrier, okay? You can flood paperwork with paperwork, okay? You can apply for 10 diseases, right? The beauty of doing this approach is you're not in love with any one of these mice anymore, right? They're scientists who just, they love their mouse model. They're going to go with this mouse model to the end of their careers. And for me, okay, we're on cancer this week, we're on diabetes the next week, right? We can just, we're very agile in that kind of way. So there's pros and cons, but you can go for whatever ethics you want before you press add to shopping cart. Other thoughts, questions?
Time just one more.
All right, it's in the front here.
You're seeing a world in which a vast amount of data is being stored for a long time, and that needs to be paid for.
That's a great question. Yeah, great issue. Who's going to pay for storing all this data? So these international repositories, a lot of ones I showed are run by the federal governments, okay? EBI kind of not so much, but the federal government, so this is, when NCBI and NIH runs this, it's really not considered intramural or extramural research. It's a product of the government in these databases, so like a function. But that may not continue indefinitely. How to keep external sources of curation, external databases going is a hard problem. It is a hard problem. How can any of us depend on these data sources if we can't actually be sure that they're going to be curated? So I think the model of the future, though, is going to be in distributing these. It's going to be mirroring these, right? Oh, you lost your side of funding, but we can keep ours going. But I also think the cost to run these repositories has to reflect real-world dollars today. It doesn't cost an Amazon.com level of investment to run these repositories anymore. There are ways to outsource the programming. I know people don't like it, okay? This is the reality. It's how the business world runs things. The cost to run repositories is going to have to come down. Expensive repositories are just, it's going to be hard to command that kind of support. A lot of times repositories run with this kind of mentality, build it and they will come. I don't think we can trust that anymore. What I would push to you, to all of you, is that if you're involved with curating the data, storing the data, looking at the data, come up with the use cases yourself. And run with it. Don't just show the world the website and the URL. Show what you did with this data. And I think that inspires people more than just numbers on a page. I have these many samples because there's so many repositories today, right? And the ones that win aren't necessarily the ones with the best user interface. It's the ones that had the best findings and the best discoveries that come from, in my opinion. I've already exceeded my time here.
I think I just about it.
Thank you very much, Hunter.